Okay, welcome to Chapter 2. This is the first of two lectures on neural mechanisms in planning and initiating movement. Nearly every system in the body is designed to support movement, but none more than the nervous system and the muscular system. The emphasis in this chapter will be on the nervous system's four main components of learning movement. They are planning, initiating, executing, and monitoring of movement. Let's take a look at the nervous system structure right now. As you can see in the diagram, there are two components to it. The central nervous system is essentially the command center, and the peripheral nervous system has components that send and receive signals from the body to the central nervous system. The sensory divisions are described as those that send signals from the periphery to the central nervous system, whereas the motor division sends signals from the central nervous system down to the muscles. As you can again see in the diagram, there is a division of messaging to muscles. The somatic system controls the voluntary movement behaviors, whereas the autonomic is involuntary and it controls more of the visceral and bodily processes. The visceral systems are part of the nervous system that represent the motor innervation of smooth muscle, cardiac muscle, and gland cells. The parasympathetic controls body functions at rest, also important when you're sleeping, whereas the sympathetic mobilizes the autonomic system and helps to provide a balance in the body by opposing the parasympathetic functions. So again, there's some balance and symmetry there. The sympathetic mobilizes the autonomic system and helps provide balance in the body by opposing the parasympathetic functions. Looking specifically at neuron structure now, we know that we have neurons throughout the body, and sensory neurons are the ones that are found in most body tissues. However, the motor neurons are found in the spinal cord. Motor neurons send messages from the central nervous system to the effectors, such as muscles or glands. The primary function of the motor neuron is to control muscle contraction. Motor neurons do not generate commands. The commands are initially received from the sensory neurons, interneurons, or brain structures like the cerebral cortex. So as I said, the heart of the nervous system are the nerve cells called neurons. And in these neurons, they have dendrites that branch out and act as receptors. Those axons carry the electrical impulse in the form of an action potential. Sensory neurons convey information from both the environment and the body toward the spine and the brain. In other words, it follows the afferent pathway or ascending pathway. Interneurons are embedded in the spinal cord and other brain structures. For example, they're housed in the central nervous system. The interneurons link motor neurons and sensory neurons with the higher brain centers. They're also considered to be responsible for aspects of the nervous system like thoughts, emotions, memory, creativity, intellect, and motivation. Motor neurons in the spinal cord are referred to as lower motor neurons, whereas the upper motor neurons are found in the brain and descend down from the spinal cord. This picture is illustrating a lower motor neuron. A motor unit equals a single lower motor neuron and its associated muscle fibers. The
In this
Association
video,
of
I want to
American
talk about
Medical
the action
Colleges
potentials
and
of
the Khan
neurons. Here we
Academy
have
have
our
put together
neuron with her soma
a
in
series
red
of
and a larger than
really
normal
nice
axon in
videos.
green, and now
And
I've
this
drawn a
particular one
myelin
speaks about
sheath
action
in yellow
potential, as I
around the
referred to in
axon,
a
and
couple
a
of
couple of larger than
slides
normal
earlier.
dendrites in blue.
So
And
let's
here's
take a
our
look at this one,
graph with
and
the
it
membrane
will remind
potential
you
on the
and refresh your
y-axis
memory a little
and
bit about
time
what
on
you
the
learned
x-axis.
in
And we've
exercise
been talking
physiology,
about in the absence
and
of input,
a little bit more
most
about action
neurons have a
potential.
stable
So have a listen.
potential across the entire membrane that I've shown here, which is often around negative 60 millivolts. And without input, the resting potential will just stay right there. But excitatory or inhibitory inputs which usually come in through the dendrites, but less often can come into the soma or the axon itself, will cause changes to the resting potential that we call graded potentials. That may be either a depolarization, also called an excitatory potential, or a hyperpolarization, also called an inhibitory potential. And these are called excitatory or inhibitory because they involve movement to the membrane potential closer to or farther away from this threshold potential, which is often around negative 50 millivolts. And we talked about how these graded potentials decay with both time and distance, so that as a depolarization from an excitatory input spreads along the membrane, the size of the graded potential gets smaller, and the same thing with a hyperpolarization caused by an inhibitory input. And we talked about how this decay with time and distance involves how much effect any of these graded potentials out here in the dendrites or the soma will have on the trigger zone at the initial segment of the axon. And that what's usually needed is multiple excitatory inputs causing depolarizations to have temporal and spatial summation, so that the size of these excitatory potentials can be large enough when they get to the trigger zone of the axon here to push the membrane of the trigger zone up over this threshold potential. And if summation of all the excitatory and inhibitory potentials at any moment in time brings the membrane potential of the trigger zone right here up over this threshold value, which is often around negative 50 millivolts, then usually an action potential will be started here at the trigger zone and conducted all the way down the axon. Action potentials being conducted down the axons of neurons are the way that neurons can transmit information over a wide range of distances, which may be 1 meter or more. Action potentials have some big differences from graded potentials in that they usually have the same size and duration for any particular neuron, and they are usually conducted the entire length of an axon basically unchanged regardless of the distance of that axon. The shape of an action potential is fairly characteristic. So it starts with summation of graded potentials getting it to the threshold value, but then instead of just decaying back down to the resting potential like graded potentials do, it has this huge rise to a very positive membrane potential where it's become more positive inside the neuron membrane than outside, which is the reverse of normal, where normally it's more negative inside the membrane than outside. After this part that's called the rising phase of the action potential, it has a short plateau, then it has a rapid falling phase back down to the resting potential. But it actually doesn't stop there. It keeps going more negative than the resting potential, then that plateaus, and then it has a bit of a slower kind of return to the resting potential from there. So the exact values may vary between neurons, but common numbers are that it might go from around negative 50, if that's the threshold potential for that neuron, all the way up to somewhere around positive 40 or so. Then it may come all the way down to around negative 70 or so before it's kind of more slowly settles back into a typical resting potential of around negative 60 or so. The total size of the action potential may vary between neurons, but for any particular neuron the size is usually the same, which is called the all or none property of an action potential. You either get an action potential or you don't get an action potential, as opposed to graded potentials where the size varies depending on the size of the input. So that if a depolarization leading to an action potential is just slightly over threshold like this, you'll get the same size action potential as a graded potential that's way over threshold. It doesn't make any difference how far over threshold you get. The size of the action potential will usually be the same. That's the all or none property of action potentials. The duration of an action potential is also usually consistent for any particular neuron. It's usually pretty quick, just a few milliseconds. Graded potentials can also be pretty fast. They can be a few milliseconds as well, but they can also be much, much longer than this. Graded potentials have a wide range of durations based on the duration of their inputs. Another big difference is that action potentials usually do not decay with distance like graded potentials do. Action potentials are usually conducted down an axon unchanged no matter how long the axon is. So that if we look right here at the trigger zone where the action potential starts, it'll have this kind of shape to its waveform. And then if we check maybe halfway down the axon, it'll have the exact same shape. And then if we check way down at the end of an axon, even if this is a meter or more, it'll have the exact same shape again. It doesn't decay with distance. The speed that action potentials are conducted down axons is often very fast. can be anywhere from 1 meter per second up to 100 meters per second. A couple of things go along with faster conduction speeds for action potentials. Larger diameter axons conduct action potentials faster than smaller diameter axons. And myelinated axons that have a myelin sheath conduct action potentials faster than axons that don't have a myelin sheath. And that often goes hand in hand. We usually see myelin sheaths on larger diameter axons. With myelinated axons, the speed an action potential is conducted down the axon is not consistent. We actually see that the action potential is conducted faster through the myelinated segments than through the gaps between the myelinated segments called the nodes of Ranvier. So that if we look here at the trigger zone, the action potential travels a little slower through there and then much more quickly through this myelinated segment. And then it travels a little more slowly through that node of Ranvier, then much faster through this next myelinated segment. This phenomenon is called saltatory conduction. Saltatory. And this word, saltatory, comes from a Latin word for jumping, because the action potential appears to jump from node to node instead of having a nice smooth conduction along the axon.
Okay, the signals in the form of action potentials can travel from dendrite to cell body to axon and then to adjacent cells or organs. And those action potentials travel through synapses, as we know, into adjacent neurons, glands, and muscles. It's important to point out that a cluster of neuron fibers enclosed in a connective sheath is referred to as a nerve. The synapses can be chemical or electrical, and in the peripheral nervous system, they're mostly chemical. And these chemical synapses are much more common and dominant in the motor control networks. Now, this diagram represents another visualization of action potential via synaptic transmission. This is courtesy of a textbook written by Edwards. Okay, let's take a look at this neuron function a little more carefully. The neuron, the transmitting or upstream end of a synapse is called a presynaptic neuron. The neurotransmitters released from a presynaptic neuron can depolarize the postsynaptic neuron, thus generating an excitatory postsynaptic potential. You'll see that excitatory postsynaptic potential here. And postsynaptic neurons is receiving a neuron downstream, and some of those neurotransmitters may also block depolarization and thus generate an inhibitory postsynaptic potential, which we can see here as well. A single neuron may form multiple synapses with another neuron or thousands of other neurons, which of course means that there's thousands of other synaptic connections. The excitatory postsynaptic potential in the postsynaptic neuron can summate to a threshold level enough to generate an action potential. The summation may be spatial or temporal and takes into account the number of EPSPs versus the number of IPSPs. I don't want you to get too caught up on the biochemical complexity of neurotransmission. It's not really the focus of this class, but I do think it's important to refresh your memory on this biochemistry, particularly from your experience in exercise physiology. If you're interested in wanting to know more, of course, you'll see in your reading on page 15, and I'll show you a quick little video on what summation is, particularly the two types that I've just referred to.
Welcome to
Okay,
another
let's take a quick
episode
look
of
at this little
Interactive
video
Biology
that
TV, where
describes
we're
a little bit
making
about
biology
summation.
fun. My name is Leslie Samuel, and in this episode, episode 19, I'm going to be answering the question, what is summation? And I'm going to talk about the two types of summation. So let's get right into it. Summation is basically the addition of postsynaptic potential. To illustrate this, I'm going to draw our membrane potential graph with membrane potential on the y-axis, E sub m, and time on the x-axis. And I'm going to start with our resting membrane potential somewhere around negative 70 millivolts. So let's say this is negative 70. Now we've spoken about threshold, and we said that in order for there to be an action potential, the membrane potential needs to reach that threshold, and I'm going to say that's around negative 55 millivolts. So an action potential comes down an axon, it reaches the axon terminals, neurotransmitters are released, and that causes an excitatory postsynaptic potential in the postsynaptic cell. What you're going to see there is there's going to be a little bump in the membrane potential. Now we know that the threshold is up here, so that bump is not going to be enough to cause an action potential. So what needs to happen is we get an excitatory postsynaptic potential, and before the first one finishes, another one comes along, raising the membrane potential even more, and that process continues over and over until eventually we have enough stimulation to cause the membrane potential to reach threshold, and then we get the action potential with our depolarization, our repolarization, our hyperpolarization, and then the action potential is finished. So that's what we're talking about with summation. We're basically adding these postsynaptic potentials. Remember, sometimes we can get excitatory postsynaptic potentials. We can get inhibitory postsynaptic potentials that bring our membrane potential even farther away from the threshold. But we're basically adding them so that we can reach that negative 55 millivolt threshold and cause an action potential. With that understanding, let's look at the two types of summation. We have temporal summation and we have spatial summation. And I'm going to do a drawing to illustrate both. With temporal summation, what we have is a presynaptic neuron. So let's just draw an axon, and I'm going to simplify it by making it just one axon terminal. And that makes a synapse with a postsynaptic neuron. So here's my neuron. Here we have the soma, and here we have the axon. And what happens is a signal comes along this axon, comes to the axon terminal, releases neurotransmitter, and that causes an excitatory postsynaptic potential in this cell. And that is where we see the first bump. Now, if it sends that signal and it sends another one quickly and another one quickly and another one quickly, and it continues doing that, that's going to cause the membrane potential to go up and up until it reaches the threshold and causes the action potential. This is temporal summation. Now that's different from spatial summation. With spatial summation, we have an axon here, and I'm just going to draw one axon with one terminal. Of course, that's simplified. That connects to another neuron. However, we also have another axon that comes, and we have an axon terminal that connects to the same neuron. And what's going to happen here is this one can cause a signal, cause an excitatory postsynaptic potential, and before that dies off, we can have another signal coming from this other cell. So we have two separate neurons causing responses in this cell, and of course we get the same result where the membrane potential goes up, and when the next one fires it goes up again, and this one can fire again, that causes it to go up, and then it eventually reaches the threshold, causing our action potential. That is spatial summation. So an easy way for you to remember this, at least this is what works for me, spatial summation is separated by space, okay, because we have two separate neurons that are firing. Temporal summation is the same neuron firing, but each signal is separated by time. So temporal summation separated by time, same neuron. Spatial summation is separated by space because they are two separate neurons. That's all the content for this video. We've looked at the two types of summation and we've answered the question, what is summation? If you have any questions, go ahead and leave a comment below and I'd be happy to get to it. That's it for this video and I'll see you in the next one.
Chemical synapses have numerous advantages over electrical ones, in particular with respect to its responsiveness and speed. The chemical synapses enable the central nervous system and the peripheral nervous system a higher degree of plasticity. And that plasticity can't be understated in terms of importance because that's where the connection between motor control and motor learning happens. Much of the work done in neuroscience is related to the importance of creating neural pathways, particularly as it relates to body movement and functionality. And subtle changes in synaptic functioning make all the difference in the world. It's that, as I said earlier, the plasticity that allows us to be able to think and adapt and ultimately change the behavior, whether it's movement or speech or otherwise. Okay, let's talk a little bit more specifically about the central nervous system and the importance of the CNS in planning and initiating motor actions. We'll talk about the peripheral nervous system in more detail later, but specifically let's discuss what it means to plan from the central nervous system. There's a number of functions that relate to the central nervous system and typically are encompassed in something called planning. And planning includes a number of different cognitive behaviors, whether it's collecting data, organizing, evaluating, retrieving memories, predicting outcomes, making all those more complex cognitive functions come to life. It's the central nervous system that's responsible for that. Planning draws upon the comparisons to previously learned behaviors and makes use of sensory feedback. And you'll recall from growth and development that the term assimilation refers to using previous experiences to learn new ones. Assimilation. The planning component of the central nervous system is really important because the seemingly simple task of initiation, which we'll talk about in a moment, requires that plan be encoded into a precise pattern of electrical signals within thousands of motor neurons so they can properly be carried out once it reaches the muscle. Now, initiation refers to having a movement plan and setting the plan in the form of neural signals out of the brain and down the spinal cord for its ultimate destination of the muscles to initiate movement. Second half of this lecture is going to discuss the brain itself. Let's first of all take a look at the idea of the cerebral cortex. The cerebral cortex, of course, is the outermost layer of brain. Its neural tissue in humans and other mammals is typically composed of folded gray matter, and it plays a really important role in consciousness. It's divided into two cortices along the sagittal plane and the left and right cerebral hemispheres that are divided by the medial longitudinal fissure. The cortex stores learned experiences and receives sensory inputs. Most of you will be familiar with the categories we've seen circled here in the different parts of the brain, and those are the ones that we're going to concentrate on today: the cerebrum, the cerebellum, the diencephalon, and the brainstem. The cortexes can be anatomically divided into different lobes, which you will recall from your anatomy class. They are anatomically and functionally divided also into the Brodmann areas, which we'll discuss further detail in a little while. The lobes, if you recall and should know by now, are the frontal, parietal, occipital, and temporal. The Brodmann areas, though, are ones that we're going to also focus on and include the motor cortex, somatosensory cortex, auditory and visual cortex as well. In this visual, you'll see a top view of the left and right cortices, and the deeper areas underneath the cortex. As I mentioned before, the cortex houses the conscious mind, and the left and right side of the brain are responsible for controlling and sensing different stimuli from the opposite sides of the body. Once again, we'll
What I've
rely
drawn
on
for you
our
here is the human brain, and this is
colleagues
what you would see
from
if you
the
were looking at
Association of
someone and they
American
were
Medical
standing in
Colleges and the
profile, or
Khan
if they were
Academy to
standing
describe it in a little
sideways
bit
in
more
front of you.
detail
So this would be the
about
front of their
the
head.
cerebral cortex.
So their
So
eyes
let
and
me
their
get this
nose and
rolling
mouth
for
would
you.
be
Let me
located
get rid
down
of that
here.
little red
This would be the back of their
thing
head.
here first. Sorry
And this
about
would
that.
be the top.
Right, here we go.
And then down here would be the bottom. And we refer to this area, this big one right here, which is the first thing that you would see if you were just looking at the outer portion of the brain. We call this the cerebral cortex. And you might notice something about the cerebral cortex right away, which is that it looks kind of wrinkly. And it actually is. Rather than having a smooth surface, it actually has a ton of bumps and folds. And this is actually extremely useful because it allows the cerebral cortex to have greater surface area. It allows it to have room for more neurons. It gives it an increased cellular mass. We usually split the cerebral cortex up into 4 different hemispheres. The first is the frontal lobe, which I'm shading in blue right here. The next area is the parietal lobe, which is back here towards the top and the back end. We also have the occipital lobe, which I'm going to color in pink. And then finally we have the temporal lobe, which I'll draw in violet right here. The frontal lobe is comprised of 2 main regions. Those include the motor strip or the motor cortex, as well as the prefrontal cortex. The motor cortex or the motor strip is responsible for our body movements. In fact, if you could peel back someone's skull and electrically stimulate different areas of the motor cortex, you could make someone's hand twitch or their leg move just by stimulating that area on their brain. The frontal lobe also contains the prefrontal cortex. And this is the part of the brain that's responsible for what we refer to as executive functions. Things like thinking and problem solving all take place in the prefrontal cortex. Not only that, but it also helps to supervise and direct all of the other areas of our brain. Another part of the frontal lobe that's worth mentioning is referred to as Broca's area. And this is a part of the brain that's associated with speech production. Let's move on to this yellowish orange portion here, which we said was the parietal lobe. And this part of the cerebral cortex is also important for a number of different tasks. One important part of the parietal lobe is the somatosensory cortex. And this is the part of your brain that's associated with feeling. And by that, I don't mean emotional feeling. What I actually mean is that this part of the brain receives information from all over your body about touch and pressure and temperature and pain. So the motor cortex would help us reach forward and grab a cup of coffee, but the somatosensory cortex is what would allow us to feel the pressure of that coffee cup or tell us how hot it is. And you might be thinking to yourself that these two things seem like they're intimately related, and they are. In fact, even though we say that they're in different lobes of the brain, they're actually right next to each other. So this side of the brain to this side of the crevice within the frontal lobe is the motor cortex. And right next to it, in the parietal portion towards the back, is the somatosensory cortex. And together they can be thought about as the sensory motor cortex. Our parietal lobe is also responsible for spatial processing or spatial manipulation. And by that I mean that it helps you orient yourself in 3-dimensional space. But this part of the brain doesn't only help us understand where we are in space, but it helps us to understand the space around us more generally. For example, if I wanted to reach out and grab a cup of coffee that's in front of me, I could use my prefrontal cortex to plan the movement and my motor cortex to complete that movement, but I also need my parietal cortex to tell me where that coffee cup is in front of me. And without that, I wouldn't actually be able to reach out and pick it up. Other things like knowing how to navigate around your house or your town, that's also what we mean when we talk about spatial manipulation or spatial processing. Our occipital lobe, which is in the back of our brain, is responsible for things related to vision. We may see with our eyes that are in the front of our head, but after collecting that information from the world around us, that information actually gets transported all the way to the back of our brain for processing. So let's put vision right here. Another term that you might hear in association with the occipital lobe is the term striate cortex. And this simply refers to the fact that if you looked at the occipital lobe under the microscope, if you looked at those cells, that they would appear striped or striated. The last lobe of the brain is the temporal cortex, and that's in violet. And this is the part of your brain that's responsible for auditory processing. As was the case with vision in our occipital lobe, information comes in through our ears, but it is processed in our brain, in our temporal cortex. Another important area in the temporal cortex is what's referred to as Wernicke's area. And when we talked about the frontal cortex, we talked about Broca's area, which we said was responsible for speech production. Well, Wernicke's area is responsible for language reception and language comprehension. Before I end this video, I just want to take a moment to point out that the different lobes of the brain do a lot more than just the things that I've listed here. However, in general, these are the things that you should think about whenever you think about the different lobes of the brain.
Okay, I mentioned earlier that the cerebrum was divided up into a variety of different ways, and one of them was the Brodmann areas. In this particular model, let me see if I can get this little gadget going again here. There it is. In this particular model, you'll see the outside area of the brain here represented by the cortexes. And we've covered this, and you've seen that on the video, of course. The inside labels, though, represent the various Brodmann areas. Brodmann areas are groups of brain cells with similar structure, and then, as a result, similar function in the body, and they're categorized or generalized by these different categories. So as we have heard earlier, visual processing occurs back here in the occipital lobe, and particularly in the visual cortex. In this diagram, we'll see the area of the brain that we mostly talk about in motor learning, which is the frontal lobe. We're looking specifically at the executive functioning that occurs and conscious thought that occurs as a result of this particular area of the brain. A lot of the planning of movement, decision making, and creative thinking occurs in the frontal lobe. Again, referred to as executive function. Although the planning happens at the front side of the brain, on the frontal lobe, it's the entire cortex that works together to ultimately execute purposeful, adaptable, and complex movements. The organization of all of that, the planning and initiation of the movement, which we'll talk about more specifically, and you've seen in some of the videos, makes it the highest level of movement control in the body.
Welcome to 2 Minute Neuroscience,
This short video
where
is going to give us a little bit more detail about the motor
topics in 2 minutes
cortex,
or less.
in particular breaks
In this
down
installment, I will discuss the
different
motor
elements
cortex.
of the motor cortex and describes a little
The
bit
motor
about how
cortex
that,
is a region of
that
cortex in the
functioning
frontal
works
lobe that is
in
involved with
terms of
voluntary
movement.
movement.
So let's
It is
roll
located
this video.
anterior to a large sulcus called the central sulcus and is often divided into two major regions, the primary motor cortex and the non-primary motor cortex. The primary motor cortex is found in the strip of cortex known as the precentral gyrus, which is located just anterior to the central sulcus. The primary motor cortex is arranged such that different parts of the region are associated with motor control of different parts of the body, and thus is said to contain a motor map of the body. Most of the neurons that travel from the primary motor cortex carrying signals regarding movement will enter one of two major motor pathways, the corticospinal or corticobulbar tracts. The corticospinal tract carries movement-related signals to the spinal cord to cause movement of the body, while the corticobulbar tract carries signals to the brainstem to cause movement of the head, neck, and face. The non-primary motor cortex is often divided into two main regions, the supplementary motor cortex and the premotor cortex. Although the functions of these areas are not well understood, it is thought the supplementary motor cortex may be involved with the execution of sequences of movement, the attainment of motor skills, and the selection of movements based on incoming sensory information. The premotor cortex contributes about 30% of the neurons that enter the corticospinal tract, but seems to be more active during the planning of, rather than the execution of, movements.
Okay, let's move on to the next slide. In the previous video, we learned about motor mapping, and what you're looking at here is something called the homunculus. It is essentially the mapping that takes place of the functionality of the motor and somatosensory cortexes and their corresponding movement functions. In all cases, muscles of the hand and face are rather overrepresented, even though they're small in number and size. They have the largest representation in the brain, and perhaps that's because through the hands and facial expressions, we're able to communicate, express emotion, and as such, we have a heightened sense of, a heightened sensory function, which requires just more region of the brain to activate and to control. In the previous videos, we've seen a little bit about the basic motor organization of the central nervous system. Essentially, the motor commands originate in the higher brain centers and then descend through the lower brain center and out to the spinal cord, exiting the spinal cord and eventually arriving at the muscles. Again, you can read more about that on page 18. The motor cortex makes the connections with the spinal cord via neuron tracts. The key tract, as we saw in the 2-minute previous video on motor cortex, is the pyramidal tract. It sends signals and messages down to the body via the spinal cord, as we've mentioned. But the extra pyramidal tract is the one here that we see that sends messages to the neck area to help with the movement required by the head. The pyramidal tract neurons are called upper motor neurons, and they work as the major controlling neurons in conjunction with the extra pyramidal tract. They make direct connections to the spinal cord and ultimately to the lower motor neurons that connect to muscle. Remember though that these tracts typically desiccate so that one side of the brain, in other words, they cross over, so that one side of the brain corresponds to the opposite side of the body. All right, let's look more carefully at the somatosensory cortex. The sensory signals here originate from the periphery, and they are the ascending signals that come up through the spinal cord. Most of these signals end up in the somatosensory cortex, which you're looking at in the diagram here. The sensory pathways are a little bit more variable than motor neuron pathways because they need to process information in a wide spectrum of brain resources. And just like the motor cortex, the somatosensory cortex makes connections with the spinal cord via neuron tracts, most of which again tend to decussate. Geez, I don't know if I'm saying that right, but you get what I mean. They cross over. Okay, I checked. I was saying that right. Decussate, the crossing over of the tracts. And as I mentioned as well, the sensory tracts are much more complex and varied, and they're often named by where they start and end. And the example I provided for you here is spinal thalamic, and you can see that spinal thalamic tract here. The somatosensory cortex is responsible for integrating a lot of different messages and signals from throughout the body. It receives information from basically every part of the body except the ears. The vestibular inputs are not considered part of the somatosensory cortex since they have their own separate category. But the collection of information that's received integrates information on pain, particularly through touch, temperature, movement, and cardiovascular function. And deep into the cerebrum, we see the basal ganglia and the corpus callosum represented here. The basal ganglia is responsible for processing information and regulating intensity of motor output. In other words, how hard do we squeeze something? What's the extent by which we deliver a force? It's highly correlated as well, the basal ganglia is, with your ability to balance. In other words, your postural control and equilibrium are connected to that particular function in the brain. The corpus callosum is really important because it links the left and right hemisphere, allowing them to communicate freely. Without that, we would have a lot less freedom in our movement and expression and ability to function normally. Neurophysiology has come a long way in the last decade and beyond since the advent of magnetic resonance imaging. It's the MRIs that allow us to be able to really understand how or what the relationship is between the activity in the brain and the movements associated with it in the body. MRI provides us with a variety of different perspectives and views. In this particular top view, each layer of the brain can be examined and is basically sliced from top to bottom, and the same thing from side to side. By now, of course, you should be able to recognize the various parts of the brain, but more importantly, do you recognize this profile and who this brain belongs to? Now, the science and physics behind MRI are beyond my capacity, but essentially we're using powerful electromagnets and the hydrogen atoms in the brain or the body to be able to look inside, essentially. Here again, another perspective of your teacher's noggin. The diencephalon includes the thalamus and hypothalamus, as well as other associated structures related to the inner part of the brain. You can see the thalamus here. It's responsible for processing information between the brainstem and the cerebrum, whereas the hypothalamus links the nervous system to the endocrine system via the pituitary gland. The diencephalon processes information that flows between the brainstem and the cerebrum, in particular receives messages and sensory signals from the visual and the somatic receptors. It's at that point where information is processed and relayed on to other destinations. The diencephalon also plays a very important role in the aspects of the autonomic nervous system. And finally, we've got the cerebellum. It sits at the base of the brain and it manages information arising from the peripheral nervous system and the motor cortex. Once it's all sorted out, it's fine-tuned and it gets sent back to the motor cortex for the execution, ultimately, of the movement plan that has been processed and developed in the brain. One way to visualize the role of the cerebellum is to consider it much like a traffic control officer at a busy intersection. This intersection in this part of the brain is where the higher command centers and the lower control functions meet. Neural traffic from other areas of the brain that is received from the effectors and muscles controlling movement passes through the cerebellum. The cerebellum plays a role in planning and evaluating and fine-tuning all motor activities. So we've come to the end of the first part of this particular Chapter 2 online learning module. Don't forget to go back and rewatch the videos if you need to, and we'll continue this chapter in Part 2 when we discuss in more detail the spinal cord in the next online learning module.